Hi, I'm Kane Park from the Center for Neuroscience Studies at Queen's University in Kingston, Ontario, Canada, and I'll be presenting work where we examine directional and general impairments in initiating motor responses after stroke. Visuospatial neglect is a neurological disorder that is commonly characterized by an inability to attend to the contralesional side of space. It has been reported in as few as 8% to as high as 80% of individuals with stroke, with a higher prevalence for those with right hemisphere lesions, but has also been reported after left hemisphere lesions, but with less severity. There have been numerous clinical tests created to diagnose visual spatial neglect, with a common one being the Behavioral Inattention Test, or the BIT. The BIT is a battery of different tests that examine visual spatial behavior. One test seen on the left here is the star cancellation, where patients are tasked with crossing out all the small stars. In this example, small stars on the top left of the test were not crossed out, which could be useful for determining whether an individual has visual spatial neglect or not. A key critique of these pen and paper tests is that they are static and do not account for time, which is a critical measure that is required to interact in our dynamic world. Studies examining reach initiation have been used to better quantify impairment in processing time for individuals with neglect. This is a study by Mattingly and colleagues. They examined reach initiation in a group of individuals with visual spatial neglect and found that reaching towards stimulus on the left took longer to reach towards than the right. Although this finding provides further evidence into impairment associated with neglect, it is characterized by only left and rightward movements. We expand on previous work by examining multi-directional reaching. On the left, we show the hand pass and speeds of a healthy control. Hand pass show relatively straight movements, and the corresponding hand speeds show relatively consistent initiation time. On the right, the average reaction time of a healthy control in each direction of reach are shown as black arrows. To simplify this 8-target reaching data, we've developed parameters to quantify reach asymmetry as well as general reach initiation impairment. Asymmetry is quantified by the reaction time asymmetry parameter, visualized as the green arrow. This value ranges from 0 to 1, with higher values indicating more asymmetric movement, and the angle of the green arrow representing the direction of asymmetry. Next, we quantified general impairment with the reaction time general, visualized as the purple arrow, which identifies the performance in the unimpaired direction. For this control, the reaction time asymmetry is directed towards the bottom right of the workspace, but the low value of 0.03 indicates their movement initiation is largely symmetrical. Reaction time general highlights a reaction time of 0.38 seconds, which can be taken together with other controls performance to determine the 95% confidence interval, and then compared with stroke patients to determine impairment. Let's look at this analysis applied to a right hemisphere damaged individual performing the reaching task with their Ips lesional arm. If we direct our attention to the left, we can see that this individual reaches each target with relatively straight movements, and on first inspection, one might interpret this behavior as normal. On further inspection, though, we can see that large variability in their leftward movement initiation, resulting in longer average reaction times. On the right, we take the average reaction time across all directions, which highlights an asymmetry towards the left, with a large reaction time asymmetry of 0.48. The purple arrow highlighting the reaction time general of 0.38 seconds is similar to the previously shown control and may indicate that this person with stroke may be performing within healthy control range and thus not have reaction time general impairment. Next, let's examine reaching across our cohort of 162 right hemisphere damaged individuals. Each blue point in the radial plot represents the reaction time asymmetry and asymmetry direction of each individual. We calculated the 95% confidence interval using 204 healthy controls, which is represented as the dashed circle, to determine the boundary of impairment. Individuals with stroke outside of this dashed circle are deemed to be impaired. Examining this figure, first you can see that right hemisphere damaged individuals can be asymmetric in any direction of reach, but across the population we see a trend for the asymmetry to be towards the top left of the workspace. Now in the figure on the right, we compare reaction time asymmetry and general to see the relationship between directional and general impairment. We can see that an L-shaped distribution where right hemisphere damaged individuals can present with impairment in only reaction time asymmetry or only reaction time general, but there are also individuals that have impairment in both parameters. About 35% were impaired in reaction time asymmetry, 32% of right hemisphere damaged individuals were impaired in reaction time general. Taken together, we identified a total of 48% of individuals impaired in reaction time. Now we move on to left hemisphere damaged individuals. For reaction time asymmetry, left hemisphere damaged individuals displayed similar results to the right hemisphere damaged, but the asymmetry was towards the top right and was less strongly correlated. Interestingly, when we look at the figure on the right, when comparing reaction time asymmetry in general, we can see the impairments in general are less impaired than asymmetry. 24% of left hemisphere damaged individuals were impaired in reaction time asymmetry, but only 12% were impaired in general. Across the two parameters, we found 30% of all left hemisphere damaged individuals had reaction time impairment. Next, we performed SROI analysis across our cohort of individuals with stroke to determine if impairment in our reaction time parameters were correlated with lesion locations. We found impaired reaction time asymmetry was correlated with lesions to the inferior longitudinal and frontal occipital fasciculus, highlighting these regions as important for processing visual information in one hemifield. Impairments in general reaction time were correlated with lesions to the superior and middle temporal gyri, which suggests this region is a key for the generation of reaching movements. To summarize, we found that individuals with stroke can exhibit asymmetrical reaction time, general reaction time impairment, or a combination of the two. Reaction time impairment is evident after both left hemisphere damage and right hemisphere damage, but is more severe after left hemisphere damage. And we also found distinct correlates for directional and general reaching impairment. I would like to thank you for taking the time to listen to this video abstract and direct you to the publication of Brain Communications for even more results as well as our interpretation of the data.